Hi everybody, this presentation is on mosquito attraction to human scent by Alicia Bingham and Heather Nelson. So to start off with, there are three common types of mosquitoes that can be found around the globe. We have the Anopheles mosquito, the Aedes aegypti mosquito, and the Culex mosquito. Some of these may be familiar to you, and many of these are disease vectors, including the Anopheles mosquito, which can transmit malaria. Alright, so let's dive into the mosquito's sense of smell and taste, or really their nose and mouth anatomy. Now, mosquitoes have not one but two noses, the maxillary palp and the antenna. They also have one mouthpiece, the proboscis. If you take a look at the picture on the right, you can find where these regions are located. And covering these organs are specialized sensilla or sensory hairs, each housing about 2 to 3 olfactory neurons. There are even about 131 different odor receptor types. Now let's look at the three major receptor types in the mosquito. The first is an odorant receptor, or an OR, and these are located in the antenna and composed of one type of receptor and the orco gene. The orco gene is required for proper dendritic trafficking to help with odorant-gated ion channel function, which in turn creates a functioning ion channel for odorant recognition when the odorant binds. The second receptor is a gustatory receptor, or a GO, and these are located in the maxillary palp and are composed of subunits G1, G2, and G3, which are necessary to activate olfactory neurons when they come into contact with a volatile or odorant, such as carbon dioxide. The third receptor is ionotropic receptors, or IR, and these are that detect odorants such as amines, acid, and ammonia. Now that I've covered both the antenna and the maxillary palp, I will just briefly go over the mosquito proboscis. Now, the mosquito proboscis consists of 6 inner needle-like structures that are surrounded by an outer sheath. The labium is the outer sheath, and this is a flexible material that can fold back when the fascicle pierces the skin. The maxilla appear twice and are needles with saw-like teeth, almost like a serrated knife that can cut through the skin. These are so sharp that you actually can't feel them probing around. The mandibles also appear twice and are used by the mosquito to hold your tissue apart after the maxilla have created an opening with their saw-like structures. This allows for easy probing by the mosquito. The hypopharynx is a stylet used by the mosquito to pump saliva into the victims. This saliva has characteristics that keep the proboscis lubricated, dilates the blood vessels, and acts as an anticoagulant. It's also considered the tongue of the mosquito. Last but not least, we have the labrum. Now, the labrum is a gutter-like stylet that acts as a probe to find the closest blood vessel within your skin. This is used to suck up the blood from the host and is actually thought to have chemosensory and olfactory cues in order to help it locate a blood vessel or vein. Now that we've talked about the mosquito nose and mouth anatomy, let's get into olfactory transduction. So odorant receptors are located on the dendrites of the ORNs, or the olfactory receptor neurons, and these are found in the small spaces formed by the sensory hairs, or the sensilla, of the mosquito. Now, the olfactory receptor neurons are mediated by a 7-transmembrane odorant receptor coupled with a G protein, as we know them, a GCPR. Now, like humans, olfaction most likely expresses a combinatorial coding strategy in which information from the receptor cells is combined when sent to higher processing. So what really happens when an odorant binds? This drawing was taken by researchers Wiebel, who demonstrates the mechanism in which mosquito olfaction occurs. Now, this pathway shows that the OR odorant receptor is lying in the olfactory receptor dendrite, and when this comes into direct contact with an odorant, the GPCR acts to activate enzymes downstream in order to synthesize second messengers that regulate ion channels carrying an transduction current, or an action potential. Now, this biochemical cascade works to then deliver olfactory information to higher level neural processing. So now that we've gone over olfaction, let's talk about taste or gustation transduction. Within the sensilla on the proboscis and also spread out on the maxillary pulp are taste receptor cells. Once these are activated, they will synapse on a gustatory receptor neuron, or a GRN. This information is then sent to the subesophageal zone into higher processing. Like humans again, it is thought that this coding system is a labeled line system, in which one receptor cell synapses only to one receptor neuron, and the information exchanged is kept separate as it travels to the higher processing center. As previously discussed, in olfaction, the ORN antennal axons are projected from the antenna to the antennal lobes and onto higher processing. In gustation, the neurons from the proboscis project to the subesophageal zone, the SEZ, and then onto higher processing. The picture on the left was taken by a light sheet microscope and allows us to visualize the olfactory receptor neurons in green found in the maxillary pulp, the antenna, and the proboscis. The picture on the right shows where the olfactory neurons are innervating the brain in specific regions. Now finally, let's just have a quick recap on the olfactory and gustatory pathways. So in olfaction, you have an odorant that will bind to an olfactory receptor. This will synapse on the olfactory receptor neuron onto the glomeruli in the antennal lobe and then to the higher processing center. In gustation, once a taste receptor cell has been activated, it will synapse on a gustatory receptor neuron, which will then move information to the subesophageal zone and on to higher processing.
Now that we have covered the approximate mechanisms, I will be getting into the evolutionary history and how the mosquito has evolved. Starting off, it is the female mosquito who drinks our blood in order to provide nutrients for her eggs. The male just feeds on nectar and will not touch us humans. How mosquitoes evolved to be bloodsuckers on humans was due to a lot of different factors, including host availability, dietary specialization, and cross-linking between different species of mosquitoes. Dietary specialization involves trade-offs between the fitness obtained from feeding on different hosts. Basically, the mosquito chooses the best host over other hosts, and even chooses the best human out of all of us. However, it took many generations for us to become the primary host. Some other factors that made the mosquito evolve to feeding on us, humans, include the amount of time mosquitoes would go searching for a blood meal. They had to choose between starving or feeding on a host that they would not normally feed on, which was aka humans. Also, the isoleucine content that is in red blood cells, which helps with the egg nourishment in females, is another factor on how mosquitoes evolved. Because in each species this content is different, and the female wants to get as much nourishment as she can. Digestion may have also played a part in their evolution due to the fact that some food or blood is easier to digest than others. There are certain mosquito species that do bite humans and favor humans over any other food source. These again being vectors of many diseases such as malaria, yellow fever, etc. They have infected many people, thus have killed many people, but not every mosquito feeds on humans. But why do mosquitoes prefer the human population is the real ultimate question. Many experiments have been done showing that certain mosquito species prefer us over species such as guinea pigs, rats, or even chickens. There are many cues and factors that drives the mosquito to our skin. Carbon dioxide is very important in their host-seeking behavior, but this alone does not distinguish humans over other species. Lactic acid being another indicator because it is more abundant in humans than other species. The main compound, however, is the AAEGOR4. You may be wondering what AAEGOR4 is. Well, it is an olfactory receptor that is upregulated in the human preferring mosquitoes. This compound, ligand, is on a female mosquito's antennae. This compound highly responds with our human odor sulcatone. I would read out the full chemistry name, but I'm not a chemist and reminds me too much of OChem. And we as humans have high levels of this. Here is the evolutionary tree showing that not all mosquitoes feed on human blood. Some do not prefer human blood at all. Some may have preferred it at one time, but not anymore, as better hosts have become available. Not just one factor contributed to human preference as stated before. There was a lot of different modalities involved as well as many other influences. I will be talking more in depth about some of these as we go along. First, let's talk about the olfactory modality and how this shaped mosquitoes into preferring to feed on humans. The very first insects lacked olfactory receptors. Olfactory receptors didn't show up until there was a first flying insect. Olfaction was very important for these newly flying insects because they were then able to detect for food and shelter. All species have a different number of olfactory receptors, which is why different species prefer different kinds of hosts. There was actually a test done showing that mosquitoes who lived in a forest environment preferred non-humans, but those who lived in a domestic environment highly preferred humans. This could be because humans are more available in domestic environments than in forests. Now let's get into the vision modality. Mosquitoes have for a long time preferred darker objects or are attracted to these kinds of objects. CO2 is a major gateway for visual search behavior in mosquitoes. Mosquitoes such as the Anopheles ignore visual objects when there's no CO2, but then become highly attracted to these visual objects after encountering the CO2 plume. Rhodopsin, which is a visual receptor protein, is non-uniform in many species, thus making species have a different host preference as well. Thermosensory and hygrosensory are also very important modalities when it comes to mosquito host preference. Mosquitoes use heat and water vapor in order to differentiate between hosts. Thus, the difference in temperature and humidity of the host tells the mosquito that the host is human, an ectotherm, a canine, etc. The thermosensory neurons are located in the mosquito's sensilla. The picture showing exactly where these neurons are located. There was a lot already said on the gustation modality. However, just to reiterate, many mosquito species are sensitive to many different compounds, thus why there is a wide range in host preference. They are sensitive to compounds on the host surface, which repels or attracts them. Now, let's talk about the plasticity and host preference for mosquitoes. Mosquitoes are able to switch hosts depending on the availability or season, or to increase their fitness and survival. CX pipiens would be an example of where it changes its feeding preference depending on the season. In the spring, early summer, it feeds on the American robin, but in the fall, winter, it feeds on humans. This is how the West Nile virus got spread. This species is also a parasitic example because they favored hosts parasitized by the parasite genus Hepatozoon in snakes than those who were not infected. Mosquitoes also learn just like you and I. We prefer different foods over others just like the mosquito. With that being said, if a mosquito has a good blood feeding experience, they learn to focus their attention on that particular host. They also prefer some humans over others as well, meaning if they like only you, they will keep coming back to you multiple times, so you better watch out. I thought this was a good picture in that it brings all the different modalities and factors together as to how mosquitoes go from choosing their host to them feeding on them if they have the right combination that the mosquito prefers. This picture also bringing together how a mosquito acts when CO2 is present. This picture demonstrates how they target and when they sense a host and how they follow the plumes of CO2 in their environment. Now let's get into another ultimate perspective, which is current function. There is a fitness advantage for females that only primarily feed on human blood. They have a greater net replacement rate and intrinsic rate of growth during their whole reproductive cycle, this being one of the main fitness advantages to just feeding on us alone, this being why humans stay a primary host for many mosquito species. I also want to answer the question as to why some people are bitten more than others, or more accurately speaking, why are you bitten more than your friend? Well, to answer this question, mosquitoes try and find humans that are not very defensive or will not feel them, and swat at them, leading to their deaths. They have evolved skills in order to distinguish between a high defensive person and a low defensive person, such that they choose hosts that contain parasites or those that give off a lot of lactic acid, because this meaning the host is physically exhausted, thus less likely to swat at the mosquito due to a slower reaction time. The more attractive cues you have, the more likely you are to be bitten. Also, the more sick and not healthy you are, the more likely you would be bitten as well.
Now, lastly, let's get into why studying mosquitoes is so important for research. This topic is so important due to the fact that mosquitoes are vectors for many of the diseases spread around the globe. And now that we know why and how mosquitoes are attracted to us, maybe we can use this information to reverse the process. If we can get mosquitoes to stop using us as a host, we might be able to stop the spread of diseases around the world. Many researchers believe that by altering the mosquitoes genetically, we can put an end to the disease epidemic. Additionally, the creation of new technology has allowed researchers to actually alter the genes in mosquitoes. Researcher Andre Cristanti has discovered a way of using the new DNA editing technique CRISPR-Cas9 to insert an infertility gene into the mosquito. However, he supports the eradication of all mosquitoes instead of just finding a way to kill the disease parasite they carry. This technique, while proving successful, is not practical because it drives the mosquito species towards extinction and must be done one little mosquito at a time. And finally, some mosquitoes have actually already been made resistant to the disease malaria. These mosquitoes were made resistant through the activation of a gene responsible for a protein that blocks this malarial infection. A British company, Oxitec, has engineered a strain of mosquitoes that requires the insertion of regular doses of tetracycline in order to survive out in the environment. So basically, they throw these mosquitoes into the environment just long enough that they can mate and reproduce to pass on these tetracycline genes. This process was able to cut a mosquito population by 80% in the Cayman Islands. However, almost every single mosquito would have to be modified in order to stop the spread of disease because if not, those infected are still breeding and still creating more infected mosquitoes. But again, this raises the question of how ethical is it to eradicate mosquitoes and their offspring? It's believed by many researchers that the only way to stop the spread of disease is to kill off the mosquitoes or make them all benign. Thanks for learning about mosquitoes. Hope you enjoyed it. And here are our works cited slides.